Good morning, dear students. This is Dr. Sabiha Khan, Department of Microbiology, Assistant Professor in the College of Barakpur, Rashtraguru Srindranath College. Today, I will be delivering a lecture on transposons. Now, we know that if we talk about transposons, there are basically two parts. First, we need to know about the transposons and the mechanism of transposition in prokaryotes, and the second one is the transposons in eukaryotes. Today, I will be delivering a lecture based on the UG honors syllabus. So we will be basically discussing today on the transposons related to the prokaryotic organisms. So without wasting much time, let's just start and enter into the introductory part of this chapter. Now, what are transposons? Transposons are the DNA segments that are found in the genomes of living organisms. These transposons are capable of moving from one place to another within a genome. Now, that means what? These are those small genetic DNA sequences that are never stable and fixed in their own positions, but whereas they keep on changing their positions. Now, they can change their positions in the same genome, they can change their positions in some other form of the genome as well. So now we come up to the introduction of this chapter. So what are transposons? Transposons are the DNA segments that are found in the genomes of living organisms. These are capable of moving from one place to another within a genome. So that means that these are those DNA elements that are never stable, and they have the capacity or capability of keep on moving from one part of the DNA to the other part. Now, it can be within the same genome, it can be within some other genome as well. These transposons do not utilize an independent form of element like the phage or the plasmids, but they directly hop from one site to another. They are found to encode a special protein called transposases. Now, this particular protein which they carry within themselves is the main protein or the enzymes that is responsible for helping the transposons to keep on hopping from one place to another. So, and this protein or the enzyme's name is transposase. This is that enzyme that catalyzes the complete process of transposition and thus helps the transposons to keep on changing their location from one place to the another of the genome. These transposons are particular to different group of organisms, and they have a major impact on changing the genetic composition of the organisms as well. It has been also found out that these transposons, they carry a lot of different kinds of genes that are related to resistance to some antibacteria, resistance to providing them the virulence and symbiotic factors and catabolism of xenobiotics. These transposons generate a variety of DNA rearrangements that have profound consequences for cell survival and evolution, and these are the basic significance of the transposons that helps to convert many non-pathogenic bacteria into the pathogenic ones and increase their tendency of virulence into a new environment where there are a lot of stress factors which actually helps the bacteria to survive more and evolve more into the new generation. These transposons are diverse in structure, they are highly regulated by a multitude of mechanisms. So finally, if we want to define the transposons, how we will be? This can be defined as a DNA sequence that is able to move or insert itself at a new location in the genome. The transposons are usually small, mobile DNA sequences that move around chromosomes with no regard for homology. We have to keep in mind, when they move from one sequence to another, irrespective of any other homology, they insert themselves into a new foreign DNA. So that is why it is mentioned here that when they move around the chromosomes, there is no regard for homology, and insertion of the transposons may produce a lot of new changes into the foreign DNA into which it is inserting. So as a result of its introduction into a new foreign DNA, it might produce deletions, it might produce inversions, chromosomal fusions, and even more complicated rearrangement. And finally, we can say that yes, the transposons introduction into a new foreign DNA actually creates a kind of mutation into the foreign DNA. That might be beneficial, that might be harmful. We will look into that later on as we go ahead. Now, before going into the details of the mechanism of transposons, the various types of transposons, we need to know the name of the famous scientist who is actually responsible for discovering these mobile genetic elements, and her name is Barbara McClintock, and she was actually awarded with the Nobel Prize for discovering this genetic instability. Basically, Barbara McClintock, she was studying with the Mendelian genetics, and she was studying with the maize seeds, the corn seeds, where she found a lot of different changes in the appearance of the corn seeds. So let us discuss into the small experimental part of what Barbara McClintock has actually done, from where those transposons or the discovery of transposons came into the science field. What she found is that there was a gene, it can be termed as capital C, which was responsible for producing purple colored kernels into the corn seeds. And that was, when it was found to be dominant, then the gene was expressed and it was expressing a purple colored kernel in the corn seeds. Now, opposite to that, when there was the recessive factor, that means this gene was not dominant, then the corn seed was found out to be carrying a white colored kernel. That means there was no coloration. Now what happened is that it was found out the presence of two transposable elements, that is the AC element and the DS elements. Both were found to be the transposons, but there were a bit differences between these transposons. That is, AC element was found to be a independent transposon that was capable of moving all by itself without the help of any other transposon from one part of the gene to another part, whereas the DS element was found to be a bit dependent type. That means it was actually lacking the enzyme called transposase, due to which, although it was a transposon, it was unable to take it out itself from its original position and stick or get inserted into a new DNA. Now, when this DS element was found to be in vicinity of the AC element, it was found out that this AC element helped the DS element to transposase itself or transpose itself, and thus it came out of its original position and got inserted into the C gene. As a result of its insertion into the C gene, what happened is that the C gene that was till long so dominant and expressing a purple colored pigment, it got mutated. So there was a change in the coloration, but as a result, again when it wanted to transpose back into its original position, that means the DS element, that means it tried to undergo a reversion. So the DS element again, it opened up from the new position and entered into its original position or any other part of the chromosome, and in doing so, it was found out that the C gene that was so mutated after reversion tried to get back to its wild type condition, but it was not able to completely go back into its wild type configuration, as a result of which it started producing the purple pigment with spots. And from here actually Barbara McClintock, she conferred or concluded that it cannot be any other genetic element but some kind of a genetic element which are actually unstable in their position, which are actually having a tendency to keep on changing their position, and it is those small elements that are actually changing their position, as a result of which a spot kind of appearance appeared irrespective of any complete coloration like either a purple or a white, but she was observing this spotted appearance. So this was a small background about how the transposons were discovered, who discovered it, and actually what happened, the genetic change exchange that was responsible for producing these genetic instability. Now, let's come up to the characteristics of transposable elements. There are various characteristics. As we will study, we will go more deeper into this chapter. Now, these transposons are DNA sequences that code for enzymes which bring about the insertion of an identical copy of themselves into a new DNA site or a foreign DNA. Transposition events involve both recombination and replication processes which frequently generate two daughter copies of the original transposable elements. One copy remains at the parent site while the other appears at the target site. Now it actually depends upon the nature of the transposons. Sometimes the original copy is also not retained at the original site depending upon the type of transposons. We will go into that a bit later. The insertion of transposable elements invariably disrupts the integrity of the target genes. As I have discussed before, once a new gene is being introduced suddenly into a foreign DNA, there will be a total disruption in its genetic characteristics. There might be a lot of changes. That means finally it is producing some kind of a mutation. The transposable elements carry signals from the initiation of RNA synthesis. For the initiation of RNA synthesis, they sometimes activate previously dominant genes also. So it depends actually what kind of a mutation it is undergoing. It might express a dominant gene, it might suppress a dominant gene, and various other factors can come into play. A transposon is not at all a replicon. You have to keep in mind, it is not at all a replicon, thus it cannot start replication. So it cannot start replication apart from the host chromosome the way that plasmid and phage can. So it is not typically like that of the plasmid DNA or the phage DNA, what they actually do during conjugation or transduction. It is totally a different story about the transposons. There is no existence of homology that exists between the transposons and the target site. So whenever a new transposon is getting inserted into a target DNA, this insertion is happening on the basis of non-site specific recombination. This is not at all a site specific recombination. Now let's come up with the various kinds of names that they are called with. The transposons can be termed as jumping genes, they can be termed as mobile elements, they can be termed as cassettes, they can be termed as insertion sequence, they can also be termed as the transposable elements or the TN elements. Now what are the types of transposable elements? Basically, if we categorize the transposable elements, they can be classified into two classes, class 1 and class 2. The class 1 transposons are known as the retrotransposons. You can see the second number transposon in the slide. So that is basically termed as the class 1 transposon, whose name is retrotransposon, and the class 2 or the DNA transposons are the class 2 transposons, which are the another class. They are totally of different category, they have different structures, although their functions are same, but their mechanism of replications are also totally different, and they exist entirely in different kinds of organisms. For example, the DNA transposons are actually in the first, you can see the first point in the slide. These transposons actually directly, they are called as DNA transposons. These are the mobile elements that transpose through a DNA intermediate and are generally referred to as DNA transposons. These are the most simplest kind of transposons that were first found out in the bacterial genes. Sometimes they are also termed as IS elements, and mostly these class 2 type of DNA transposons are found in the prokaryotes. Now coming up with the retrotransposons. The retrotransposons are those that transpose via an RNA intermediate that are transcribed from a mobile element by an RNA polymerase and then converted back into a double-stranded DNA by a reverse transcriptase. As we all know, what is the mechanism in which a retrovirus, they actually replicate themselves. So this is what actually happens in the retrotransposons also, and they must have an RNA intermediate, they must have RNA polymerase, and they must have the reverse transcriptase enzyme, with the help of which actually they can synthesize their cDNA and finally create a double-stranded DNA. They are called retrotransposons because their movement is analogous to the infectious process of retrovirus. It is just typical to the type of replicative mechanism that the retrovirus actually undergoes. Now, I have already discussed the type of transposons. I would like to categorize these transposons again on the basis of their mechanisms in which they are replicating. Firstly, we have classified the transposons on the basis of their structure, the structural aspect, and as well as into their existence. That is, some of them they exist into the prokaryotes, and some of them they exist into the eukaryotes. Now, depending upon the type of transposons which is replicating in which mechanism, we can divide them into cut and paste transposons. We can divide them into another second category as the replicative transposons, and the third category is the retro elements. Now, basically, before going into the mechanisms, I would just like to mention 2 points. That is, when the transposons are actually duplicating themselves or replicating themselves, there can be actually any 2 mechanisms. It can happen that the transposon, when it is trying to cut off from one side and get integrated into a new position, it might not replicate itself. It might not leave a copy of itself and straight away come out in its original form from the original position and get inserted into any other part of the DNA of the either of the same genome or separate genome. So it is the way it is doing, it is known as cut and paste. That means simply the transposon is cutting itself out without leaving a copy of itself and then pasting into its new position. This is one kind of a mechanism, and another mechanism is that it tries to keep or leave behind a copy of itself. That means before coming out, it will try to replicate, increase in its number, or rather duplicate, leave a copy of itself, and the new copy that has been produced after replication, that copy gets inserted into the new DNA. And retrotransposons or the retro elements are, as I have discussed a bit in just the previous slide, it is some kind of a retro element where retro is similar to the retroviral replicative mechanism carrying the reverse transcriptase gene. We will be going into the details of all these mechanisms. Now, firstly, I am discussing the most simplest kind of mechanism of the transposons or the transposition mechanism, which is the cut and paste mechanism of transposition. Please try to concentrate into the right hand side of the slide, that is the diagrammatic part. The diagram entirely will help you understand that what mechanism is actually being followed for the cut and paste mechanism. So it is non-replicative. That means what? Its tendency is not to keep a copy behind. Let's see the first structure of the DNA transposon. It is basically the DNA transposons that undergo this kind of mechanism. So we have the light green color part that actually is responsible for the gene producing the transposase enzyme. Then we have the terminal inverted repeats. The transposons characteristics are like this. These are the typical structure of a transposon, where they have the central part carries the transposase gene, that it is flanked by two terminal inverted repeats, and you can see the gray colored part, which is the flanking host DNA in which it is present. So this is the original DNA transposon, how it looks like. Now, the transposase, it's an enzyme that is being produced from this light green colored gene part. So this transposase gene product, or the enzyme that is being produced, it is usually a 2 or a 4 subunit enzyme. What is this enzyme? This is the only heroic enzyme that actually helps the transposon to cut off from its original position and helping it to get inserted into the new DNA. So the complete action is carried out only with the help of this transposase, which is actually present only in the AC kind of transposons, or the AC element kind of transposons, or the unstable independent autonomous transposons. These are found to be absent in the DS elements, which are the unstable dependent transposons. So coming up to the enzyme transposase, as I said, these are either 2 subunits or there are 4 subunits. Now what they do? The transposase enzyme, they bind directly to the terminal inverted repeats. That means through the 2 dark green terminal inverted repeats, the enzyme is being produced and it is binding, and as a result of its binding, you can see the orange colored thing, which is actually the enzyme, and this is binding to the 2 dark green parts, or the terminal inverted repeats. So as a result of this binding, the transposon is pulled a bit closer to each other, which is actually forming a horseshoe kind of a structure. So there is a small bend into the structure. Now this brings the 2 ends together. Stable protein DNA complex is formed, and this complex is called as synaptic complex or transpososome. Okay. Now this transposase, after this binding, come to the third diagram, or the third step of the diagram. Now this transposase, what it does, after binding, its job is to cleave, because its target is to take it out, just extract it out. So what it does is that it will do the cleaving. Now this cleaving never happens in a single step. What I mean to say is that the cleaving will happen firstly in the 3', so that it can create a 3' OH. That means in the 2 strands, firstly the cleavage occurs so that a 3' OH is created, and then on the successive steps, the 5' phosphate is also cleaved. Now there is actually a kind of a complicated mechanism that you need to understand, but because I am delivering a lecture for the UG honors students, you really don't need to get involved with that complications right now. When I will be discussing with the PG part or the postgraduate part, then I will be going into the details of this mechanism. There is a bit of complex mechanism onto how this phosphate or 5' phosphate strand is created as well. Anyway, so what you need to know is that there is a cleavage, and finally the transposon sequence that is coming out with the free 3' OH group at each end. Now this is actually the main target. If I want my transposon to come out of its original position and get inserted, so in order to get inserted into a new DNA, it must have a 3' OH end. That is the main target. So the target has already been reached. It's successful in creating a 3' OH group on both the ends of the transposon. So what happens then? Other DNA strands cut by various, as I said, the transposon, finally it's getting excised. That means it's coming out. Okay. Now, the 3' OH end of the transposon DNA attack the DNA phosphodiester bonds at Let's come up to the first, second, third. Come to the third part of the diagram given in the right hand side. Okay. Now you have the 3' OH. What will it do? It will go and attack the phosphodiester linkage of the new target DNA. So NICs are introduced in the other target DNA strand, few nucleotides apart. Transposons are joined via a reaction. They are called as DNA strand transfer. After this, few nucleotides between NICs, they are left in the small stranded gaps. As you all know that this is already done, it's the same mechanism as it is followed in the replication pathway. So there is a NIC sealing, finally the DNA ligase comes into play and this does the final sealing and there is no more discontinuity. The new target, new DNA or the new transposon has already entered, and if there are any kind of a double-stranded break, that will be repaired by the homologous recombination mechanism, and finally the new DNA that you are getting contains the new target DNA, contains the new transposon that has been inserted very successfully. So this was a kind of a small view about how the non-replicative cut and paste mechanism is actually occurring. Now we will go a bit deeper and a bit complex mechanism. The cut and paste mechanism of the transposition was a bit easier one. That usually happens in the prokaryotes. Now, if we go into the eukaryotes, basically this mechanism is a bit complex and it usually involves, many prokaryotes also undergo these replicative mechanisms as well. And what they have is that most of the circular DNA, mostly the circular DNAs, they undergo this kind of a replicative transposition. So what its target is? It has to duplicate itself. So there is one particular kind of a transposon. You can see in the diagram, please concentrate into the right hand side of the diagram. You can see the first step of the diagram where it is written as donor DNA. There is light green and the dark green part, which is the transposon. It is present in the donor DNA. Now this transposon needs to get replicated first, and then the newly replicated one that is produced, or the daughter transposon that is, or the daughter DNA that is produced, that will now get inserted into the target DNA. So what happens? The transposon DNA replicated during each rounds of transposition. Transposase assembles on each end of the transposon to form a transposase, as I've discussed earlier. This transposase introduces NICs at junctions between transposon and planking host DNA, thus generating a 3' end or OH end on the transposon. But at this position, the complete excision of the transposon is actually not happening. So what is happening is that the original donor DNA, when it is attacking with the help of a transposase enzyme and carrying the transposon, when it is attacking the target DNA, it is getting attached to the target DNA, and finally it is forming a kind of a complex which we call it as a co-integrate. Come up to the 1, 2 and the third part of the diagram, where see, you can see the donor DNA, which is gray in color, is actually getting attached to the target DNA. In the previous mechanism, the donor DNA had no relation in getting attached, but here they are getting attached because at this point there is no replication of the transposon that has actually occurred. Okay, so carrying only the one set of transposon, the donor DNA is attacking with the help of the, obviously with the help of the transposase enzyme, by creating a 3' OH, as you can see in the second part of the diagram. And with this 3' OH, it is actually attacking the target DNA, and thus it is getting attached and forming a complex which we call as co-integrate. Fine. Now, this 3' OH is joined to the target DNA by strand transfer reaction, and after this, After this, the same process is actually happening that has happened into the replicative mode or the replication cycle, and on replication the transposon gets replicated or doubled, and this kind of come up to the last diagram of the co-integrate with two copies of the transposon is formed. There is nick sealing, there is ligation, there is recombination if there is any kind of a double-stranded breakage, and finally this co-integrate is actually, you can see it is carrying the two copies of the transposons, and as well as the donor and the target DNA, they are also attached to themselves, and after this they are again separated from each other, and the donor carrying its own DNA transposon gets separated with the target, which now has or it is able to carry a new copy of the transposon. Now, so the replication proceeds through transposons and it stops at the second fork. Two copies of the transposon flanked by short target site duplications, and this frequently causes, sometimes due to this it can lead to chromosomal inversions and deletions or any other detrimental effect to the host. Okay, so this was a small view about the two mechanisms of how these transposons can replicate, either in a non-replicative mechanism or in a replicative mechanism. So if I again go back to the categorization of the transposons, we can categorize them as DNA transposons that are mostly using cut and paste replicative mechanisms, the viral-like basically the retrotransposons, they can be of two types, the viral-like retrotransposons and the poly-A retrotransposons. Both are retrotransposons. Now let's concentrate again into a bit of structure, okay, so that you people don't get confused. So the easiest DNA transposon or the IS elements, as I have discussed it earlier, you can see the right hand side of the slide, the first diagram, that is A DNA transposon. What is it carrying? It is carrying a transposase gene, two terminal inverted repeats on two sides, and then you have the target site duplication with which it has attached due to the process of, or with the help of the process of recombination. So this is a very simple structure that a DNA transposon is actually carrying, and starting from one terminal inverted repeat to the another terminal inverted repeat, we would be calling this total thing as the element or the transposon. Now come up to the second kind of a retrotransposon, which is the viral-like, or mostly the retrotransposons or the transposons that is being present in the retrovirus. So these are viral-like. What it carries? It carries two genes that code for two proteins which are known as integrase and the reverse transcriptase, without which it cannot transposase or cannot convert into a double-stranded DNA. So in place of this transposase, the viral-like retrotransposons are carrying integrase and the reverse transcriptase. Okay, and then we have the LTRs, and you again have the target site duplications. Okay, now coming up to the poly-A retrotransposons. Poly-A retrotransposons, they have a some kind of a similar structure with the retrotransposons, but they do have the non-viral part, and they also undergo replication with the help of RNA intermediate, but they also have a poly-A tail. So what they have is, they have a 5' UTR and a 3' UTR at the other end. What is this UTR? UTR is the untranslated region, and then they code for two enzymes, or they have two genes, basically the ORF1 and ORF2, that code for different enzymes that we will be going in details when we will be doing the poly-A retrotransposons. Okay. Now coming up to the viral-like, here the integrase plays more or less the same part or same role as that of the transposase in the DNA transposons, and you know the activity of reverse transcriptase, what it does. So now we will be starting up with the class 1 transposon or retrotransposons. These retrotransposons can be subdivided into two groups. that are distinguished by the presence or absence of a long terminal repeats or the LTRs. So that means depending upon the type of LTRs, we can divide the retrotransposons in the two forms. That is, one, the LTR retrotransposons, that means those retrotransposons that are carrying the long terminal repeats. And the second category of retrotransposon is the non-LTR or the retrotransposons that do not carry the long terminal repeats. Okay, there are very good examples of these retrotransposons that you will be dealing very soon in the other part of my delivery of the lecture. That is, the human LTR elements are the endogenous retroviral like, that is the HERVs one, and most of the non-LTR retrotransposons are those, as we all know, the line and the sign and the ALU sequences that we deal with them a lot. Now, coming up directly to the viral-like or the virus-like retrotransposons and the retrovirus. So what they do have? They have the reverse transcriptase. What is this? This enzyme is that uses the RNA template. So without the help of the RNA template, it will not be into action. So wherever there are the RNA templates, these reverse transcriptase enzymes, they help them to convert or convert the RNA into a cDNA or a complementary DNA. The retrovirus genome is packaged into viral-like particles, leaves the host cells and infects a new cell. The retrotransposons can only move to a new DNA sites within the cell. So let's come up into the mechanism of how these viral-like retrotransposons and the retroviruses are actually duplicating or transposing. Please concentrate on the right-hand side of the slide. So retrotransposons, DNA transcribed into RNA by host, RNAP transcription starts at promoter with LTR. Okay, you have the LTR, you have the promoter region with which the RNA polymerase will bind. A transcription method, just in the plain method how the transcription is happening, it will produce an RNA intermediate. Now this RNA intermediate is sufficient, which is actually carrying this transposon element or the TN element, and with the help of the reverse transcriptase enzyme, the reverse transcription is actually occurring and it is producing a cDNA, a double-stranded DNA. So the RNA reverse is transcribed by the reverse transcriptase to produce a double-stranded DNA, which we call it as the cDNA. Now comes the role of the enzyme integrase. It has the similar action as that of the transposase enzyme present in the first class, that is the DNA transposon. So this integrase, it recognizes and binds the ends of the cDNA and then cleaves. That means the job of the integrase or the transposase is just to bind and put a cut, cleave, create a 3' OH, nothing else. Okay, so this is what it does. It is creating a 3' OH. Please check into first, second, third, fourth step of the diagram. So the dinucleotide has been released, a 3' OH has been created on the two ends of the cDNA. Fine. Now, the integrase performs strand transfer reaction. That means what? The moment you are getting a 3' OH, it is ready to get inserted into a target DNA. That is what it is going to do. The 3' OH, please concentrate into the first, second, third, fourth, fifth step. Come to the fifth step where the 3' OH is directly attacking to the phosphodiester linkage of the new target DNA irrespective of the site. Fine. So the same process is happening again. I will be not discussing the rest of the steps which we have discussed earlier. So the attack has occurred, the new DNA has entered here, then nick sealing, the ligation will happen, gap filling will happen, and any kind of a recombinational event, if it is required, will happen, which will finally convert or produce a new integrated copy of the target DNA, which is now carrying a new transposon. Okay, so this was the mechanism by the help of which the viral-like retrotransposons usually do. Now coming up to the transposable elements that are found, that is to the transposable elements that are found in prokaryotes, or coming up to the class 2 transposable elements. They can be categorized into two types. The basic kind is the IS element or the insertion sequences, and the other kind is the prokaryotic transposons with a little bit of variation in the insertion sequences. So come up to the insertion sequences. Please concentrate into the lower part of the slide, where you can see there are various kinds of insertion sequences, and they have been named with different numbers. They have been basically isolated from the bacteria, and they can be named as IS1, IS2, IS3 and so on. I'll be discussing here just the plain IS elements structure. So what is this? It is a simplest type of a transposable element found in bacterial chromosomes and plasmids. What they do? They encode the transposase gene, which is which is responsible for mobilization and insertion. Their size can range. Usually they are smaller in size, but their size can range from 768 base pair to 5 kilo base pair. The first identified IS element is called as IS1, that has been identified from E. coli. It has been identified from the E. coli's galactose operon, and it is 768 base pair long and is present within 4 to 19 copies in the E. coli. That means one particular IS element is present either with the 4 or 19 copies. There are several copies of these transposons that are found in the E. coli chromosome. The ends of all the IS elements have the inverted terminal repeats, or the ITRs or the IRs. Okay, so that is the structure of the basic insertion element or the insertion sequence. Now we will be discussing about the different, there are a lot of variations into these prokaryotic DNA transposons. So depending upon these kinds of variations, they can be classified as the composite transposon or non-composite. These are the other categories of the transposons that are also found in the prokaryotes. So what is a composite transposon? Composite transposon is similar in function to simple transposon and insertion sequence elements, in that it has a protein coding DNA segments flanked by the inverted repeats that can be recognized by the transposase enzymes. Composite element carries that. A composite transposon, however, after all these, carrying all these, it is flanked by two separate IS elements. Actually, basically, if we combine the two separate IS elements, so that converts into a composite one. So composite transposon is nothing but a combination of various IS elements. So that is what is written here. A composite transposon, however, is flanked by two separate IS elements, which may or may not be exact replicas. That means a composite element can form with a combination of two IS1 elements. Sometimes there can be a composite transposon with a formation of one IS and one IS3 element. So depending upon the type of composite transposons, the combination can happen. And instead of each IS elements moving separately, the entire length of DNA spanning from one IS element to the other is transposed as one complete unit. So the composite transposons will also often carry one or more genes that are responsible for conferring any kind of an antibiotic resistance. This is the main speciality about the composite transposons, actually, that they are a combination of the IS elements, and together with the combination, one of the IS elements may be inactive in producing the transposase gene, but whereas the other element, the other one is active, it is producing not only a transposase gene, it also carries a antibiotic resistance gene factor as well, which is able to provide a new resistance or a characteristics of providing a antibiotic resistance to a particular antibiotic. Okay, so you can see the structure, how it looks. It carries the transposase as well as it is carrying here it is, you can see it's a TN10 element. The composite transposons are named separately. Once the IS elements are combining and a new composite element is forming, so then they are termed as TN elements. So there are various kinds of composite elements. I am discussing here with the example of the TN10 element, which consists of two IS10 elements, that is flanking a gene of tetracycline resistance gene, and they do also have these inverted repeats as well. And what are the non-composite transposons? These transposons do not contain IS elements flanking to the protein coding genes. Okay, they contain certain repeated sequence at both the ends, and they also contain certain other genes that are now not actually required for transposition. So they are transposons, but together with they are not always involved in carrying the genes for only causing transpositions. They are also carrying some other extra genes which are not at all related in the mechanism of transposition actually. So the protein coding genes may code for some kind of a drug resistance. Okay, so a very common example of the non-composite transposons are the TN3 and TN21. This is a small diagrammatic representation where it is carrying a TN3. It is a 5 kilobase pair. It is carrying a transposase gene. It is carrying a repressor gene. It is carrying a beta-lactamase gene. So what happens is that these non-composite transposons, these are they lack the flanking insertion sequences, but this transposition requires some specific DNA sequences that are located at the ends or in between the multi-subunit complex, and they encode the transposition proteins definitely, but they have also the inverted sequences at their ends with no IS elements. And in addition to the resistance gene, they also provide genes which can mark some special characteristic like the virulence gene, any other drug resistance gene, and so on. So coming up to the last part of my today's lecture, we will be concluding, or we will be giving a small diagrammatic or a chart form difference between a composite and a non-composite. So you have known composite transposons are transposons that terminate with the IS elements. They do not have IS elements. These have the insertion sequence. They do not have insertion sequence. And you can take the examples. Some which is TN10 is one of the examples of a composite transposon, whereas TN3 and TN21 is the example of a non-composite one. Before we end up, I would also like to give you a small view about the mu transposon. Okay. Now, mu transposon is one of the robust kind of a transposons. We know usually the transposons are very small in size, but mu transposon is found out to be that kind of a transposon which carries a very long base pair. So it's a very huge size. Okay, so this transposition actually requires two phage encoded proteins. That means it requires two important genes or the proteins which is important for it to transpose. One is the transposase, which is encoded by a gene called gene A. I will show you its structure. And another is the transposition enhancer, which is encoded by another gene in its structure, which is called as the gene B. Okay, now in bacterial cells, this mu is actually a phage. So this mu phage transposition can be non-replicative, leading to a lysogenic mode of lifestyle, or it can be replicative, leading to a lytic phase. Now here is the small diagrammatic sketch of how it is producing this transposon is actually getting transferred to the target site. So there is a nick, and again it is nick, and then it will form a co-integrate complex. See, it has formed a co-integrate complex, and that is how that has entered into a new target DNA. Now mu phage or the mu transposon has got a very special characteristic due to which it is famous, and that is why I am discussing here. before I complete my delivery of the lecture. That means mu-phage has got a very important role, that is, it is involved in not only transposition, but it is also involved in regulating itself. That means it has some kind of a mechanism with the help of which it can regulate its copy number. That means a transposon cannot keep on multiplying and changing its position and continuing. So if it happens this way, this will never be controlled. This will go out of control. So that has to be regulated. That means how many copy number of transposon is to be produced, or how many times it is going to get transposed, that is actually also regulated. So the mu is one of the examples. There are also other examples. You do not need it in your honors level. So I'm just trying to throw a little bit of light into this mu thing, and this mu uses target immunity to avoid transposing onto its own DNA. So it is trying to produce a kind of an immunity so that its own transposon will not enter into its own DNA. It will rather enter into a foreign DNA. Okay, transposition in its own genome causes disruption in its gene, and this disruption is actually solved by target immunity. This is actually achieved by the two proteins which is known as mu A and mu B ATPase. The mu A inhibit mu B from binding to the nearby DNA sites. Actually, the mu A is inhibiting this mu B, and this inhibition actually requires ATP hydrolysis, as a result of which mu A and mu B helps mu A to find a target site for transposition, and then it gets transposed into a new DNA. So if we conclude into the complete part that I have discussed today, transposons cause major changes in structural and functional aspect of the entire genome. Transposons cause various kinds of mutation as deletion, insertion, etc. Tremendous impact in altering the genetic organization of the organism. It can completely alter the genetic organization of an organism, converting into completely a new organism. Transposons mediated gene tagging is done for searching and isolating a particular gene. It is very useful as cloning vectors, and it is also useful as genetic markers in genome mapping. So this was all for today's lecture. Thank you all.